Research performed at the Silvers Lab is related to the areas that are commonly referred to as structural biology and molecular biophysics. In other words, our objective is to understand biomolecular systems and their biological function by studying their molecular structure, structural organization, and their dynamic behavior at varying levels of complexity. Fundamental research into how biomolecules work on a molecular level is very important. Once we understand these molecular level events, we can connect the dots between the molecule's behavior and its function, and maybe more importantly, its dysfunction. This way, our studies directly relate to understanding diseases such as cancer and neurodegenerative diseases such as Alzheimer's, as well as lay the groundwork for the development of drugs and therapies combating these diseases. One line of research in our lab is to understand how certain proteins interact with RNA and how this interaction is involved in post-transcriptional gene control that regulates the delicate balance between active translation, degradation, and storage of mRNAs. Many of these protein-RNA interactions are involved in the onset of various cancers and neurodegenerative diseases, as well as fibrosis. Our method of choice to achieve these objectives is nuclear magnetic resonance spectroscopy, or short NMR spectroscopy. In NMR spectroscopy, we expose our biomolecule to a strong magnetic field by placing it into an NMR spectrometer. Biomolecules are made out of atoms that are arranged in a very specific way, and in NMR spectroscopy we can use the nucleus of most atoms as reporters. To achieve that, we manipulate the nuclear spins within our biomolecule by sending in radio waves to excite these nuclei and then detect their response with sensitive radio receivers. Every nucleus has a unique position in our biomolecule, and every nucleus has a unique chemical surrounding. Hence, the data we collect informs on the local structure and dynamics of our biomolecule. This way, we can not only study the three-dimensional structure of a biomolecule, but also the molecular motion on a multitude of timescales. In our lab, we control the entire process from start to finish. We make our samples, including proteins, RNA, or DNA, and perform NMR experiments and analyze data. We have access to departmental NMR spectrometers, as well as NMR spectrometers located at the National High Magnetic Field Laboratory in Tallahassee, the largest magnet lab in the world.